Welcome back to daily lectures by New Age Cosmetic White EdTech Healthcare Consultancy. I'm Dr. Bobby. We run the subject of molecular biotechnology, and on the specific chapter of commercialization of proteins. To put it more specifically, synthesis of commercial products by recombinant microorganisms. We are at the subtopic of design antibiotics. Well, in recent years, there's been an enormous proliferation in the prevalence of antibiotic resistance bacterial infections. At present in the US, more deaths are attributable to infections by methicillin-resistant strains of Staphylococcus aureus than to human immunodeficiency virus, or AIDS. An important component of S. aureus virulence is the carotenoid pigment staphyloxanthin that is synthesized by the bacterium. As a consequence of the large number of conjugated double bonds that this compound possesses, it can detoxify the reactive oxygen species that are produced by the host immune system in response to the bacterial infection. On the other hand, strains of S. aureus that do not contain staphyloxanthin are rapidly inactivated by the reactive oxygen species produced by host neutrophils. This observation suggests that the disruption of S. aureus staphyloxanthin synthesis might be a suitable target to prevent the proliferation and toxicity of the bacterium. And the first committed step in the biosynthesis of S. aureus staphyloxanthin, the enzyme of dehydrosqualene synthase, condenses two molecules of farnesyl diphosphate to produce presqualene diphosphate. In S. aureus, this compound is modified to yield dehydrosqualene, which is converted into 4,4-diaponeurosporine. Let me repeat that for you: 4,4-diaponeurosporine, and eventually into staphyloxanthin. Interestingly, the synthesis of cholesterol in humans also proceeds through presqualene diphosphate in order to determine whether the S. aureus enzyme that catalyzed the first committed step could be a target for a design antibiotic. The bacterial gene was cloned and overexpressed. The enzyme was purified to homogeneity, and its X-ray crystallographic structure was determined to 1.58 A resolution based on the three-dimensional structure of the enzyme, as well as the presence of inhibitors of the human enzyme from the cholesterol synthesis pathway that performed the same function. Based on the three-dimensional structure of the enzyme, as well as the presence of inhibitors of the human enzyme from the cholesterol synthesis pathway that performed the same function, a series of 8 chemical inhibitors was designed, synthesized chemically, and tested. 3 of the inhibitors, which when tested at the levels below 1 microgram, dramatically blocked the conversion of farnesyl diphosphate to presqualene diphosphate, which is highly pigmented and therefore easy to visualize. In fact, one of the best inhibitors turned out to be a drug candidate that had already undergone preliminary testing in humans for its ability to lower cholesterol levels. Despite the fact that these results are preliminary, the compound that was selected using this strategy caused 98% decrease in surviving S. aureus in infected mice. Thus, while much remains to be done, this work demonstrates that by choosing a non-conventional target, it is possible to develop design antibiotics that render pathogenic bacteria susceptible to human and animal immune systems. Biopolymers. Biopolymers are large multi-unit macromolecules synthesized by microorganisms, plants, and animals. Some of these polymers have physical and chemical properties that are useful to the food processing, manufacturing, and pharmaceutical industries. The ability to genetically engineer organisms has stimulated researchers to design new biopolymers, replace synthetic polymers with biological equivalents, modify existing biopolymers to enhance their physical and structural characteristics, and found ways to increase the yield and decrease the cost of biopolymers produced by industrial processes. Xanthan gum, Xanthomonas campestris is a gram-negative obligatory aerobic soil bacterium that produces the commercially important biopolymer xanthan gum. A high molecular weight exopolysaccharide as a byproduct of its metabolism. This polymer has a cellulose backbone made up of a straight chain polymer of glucose units. Each of its trisaccharide side chains includes one glucuronic acid and two mannose residues, which are attached to every second glucose residue of the backbone. A xanthan gum has a high viscosity, stable in extreme physical and chemical environments, and exhibits physical and chemical properties similar to those of a plastic. In particular, its physical properties make it useful as a stabilizing, emulsifying, thickening, or suspending agent for successful commercial products of xanthan gum. X campestris should be grown in an inexpensive and plentiful carbon source. Wild-type X campestris can efficiently utilize glucose, sucrose, and starch, but not lactose as a carbon source. Whey is a waste byproduct of the cheese-making process that consists of water, 94 to 95%, lactose, 12.5 to 4%, and small amounts of protein, minerals, and low molecular weight organic compounds. Enormous quantities of whey are generated by the dairy industry, and its disposal is a major problem. In North America, whey has been used extensively as a filler in the preparation of prepared foods. However, with increasing awareness that large numbers of individuals are lactose intolerant, it is imperative that alternative uses be found for this material. Moreover, disposing of whey by releasing it into rivers and lakes can deplete the amount of available oxygen, thereby killing many of the organisms. Transporting whey to landfill sites is exceptionally expensive, and potential groundwater contamination by the discarded whey is a major concern. So let's read that again. Whey is a waste byproduct of cheese-making process. It consists of water, 94 to 95%, lactose, 3.5 to 4%, and small amounts of protein, minerals, and low molecular weight organic compounds. Enormous quantities of whey are generated by the dairy industry, and its disposal is a major problem. In North America, whey has been used extensively as a filler in the preparation of prepared foods. However, with increasing awareness that large number of individuals are lactose intolerant, it is imperative that alternative uses be found for this material. Moreover, disposing of whey by releasing it into rivers and lakes can deplete the amount of available oxygen, thereby killing many of the aquatic organisms. Transporting whey to landfill sites is exceptionally expensive, and potential groundwater contaminant by the discard of whey is a major concern. Finally, the costs of removing the solid component of whey are prohibitive. Consequently, many schemes have been devised to use whey in creative ways. Theoretically, whey could be used as a carbon source for growing industrially important microorganisms. With this in mind, X campestris was genetically engineered to grow on whey. The E. coli LAC-ZY genes, which encode the enzymes beta-galactosidase and lactose permease, were cloned onto a broad host-range plasmid under the transcriptional control of an X campestris bacteriophage promoter. This construct was introduced into E. coli and then transferred from E. coli to X campestris by tripartite mating. Transformants maintain the plasmid, express the enzymes beta-galactosidase and lactose permease at high levels, utilize lactose as the sole carbon source, and produce large amounts of xanthan gum with glucose, lactose, or whey as the carbon source. By contrast, wild-type X campestris produces large amounts of xanthan gum, only when grown on glucose. The system may well be able to convert a nuisance waste product into a substrate for the production of an economically valuable biopolymer. Okay, so that's pretty interesting and an important piece of information for future research into this subject. Melanin. Melanins are a large, diverse family of light-absorbing biopolymers that are synthesized by animals, plants, bacteria, and fungi. It has been suggested that these pigments might be useful as topical sunscreens, sunlight protective coatings, for plastics or additives for cosmetic products. Currently, melanins are obtained in small quantities either by extraction from natural sources or by chemical synthesis. However, recombinant DNA technology has made it possible to produce a range of melanins with different physical properties inexpensively and on a large scale. Biochemically, melanins are irregular, somewhat random polymers that are composed of indoles, benz-thiozoles, and amino acids. The first step in their synthesis, which is catalyzed by the copper-containing monooxygenase tyrosinase, is the oxidation of tyrosine to dihydroxyphenylalanine quinone. The final stages of the polymerization of melanin are non-enzymatic, and depending on the chemical nature of the non-quinone components that are incorporated into the polymeric structure, typically hydroxylated organic compounds, the end product can be black, brown, yellow, red, or violet. The genes involved in melanin biosynthesis and the bacterium streptomyces antibiotics has been isolated and analyzed. These genes were selected from a clone bank of S antibiotics DNA on the basis of the ability to change color in the presence of specific components that were added to the medium. They consist of two open reading frames, ORFs, open reading frames, open encoding tyrosine molecular weight 30,600, and one ORF 438 encoding a protein of unknown function with a molecular weight of approximately J4800. To test whether both of these genes are required for melanin production, they were subcloned into an E. coli expression of vector where one construct contained only the tyrosinase gene and another carried both the tyrosinase and OR438 genes. The vector with the tyrosinase gene directed the synthesis of a large amount of tyrosinase than did the vector containing both the tyrosinase and the OR438 genes. However, the amount of tyrosinase was relevant because it turned out that melanin biosynthesis required the production of both genes. The protein encoded by OR438 may act as a copper donor to apotyrosinase, an active precursor form of tyrosinase. Apotyrosinases are activated by acquiring copper ions under natural conditions after dihydroxyphenylalanine quinone is produced by tyrosinase. A variety of low molecular weight compounds, non-quinones, can be incorporated into the final polymer. The chemical and physical nature, including the color of the melanin that is formed after cloning of the key genes into E. coli, may be manipulated to some extent to form melanins with different properties by the addition of different amounts of specific low molecular weight compounds to the medium. Adhesive protein. Researchers are trying to inexpensively produce an adhesive protein biopolymer originally isolated from the blue micelle Mytilus edulis in microbial cells. This biopolymer is an exceptionally strong waterproof adhesive protein called bicyl adhesive that enables the muscle to attach very tightly to a variety of surfaces. Following its secretion, the bicyl adhesive becomes highly cross-linked randomly, and consequently the protein cannot be sequenced. Without this information, it is impossible to deduce nucleic acid sequences that might be used for the synthesis of DNA hybridization probes. However, it was possible to isolate an intracellular precursor form of the basic protein called the 130 kilodalton precursor protein. Production of 2-keto-L-gluconate, an intermediate in L-ascorbate synthesis by a genetically modified Ovenia herbicola. Courtesy S. Anderson, C.B. Marks, R. Lazarus, J. Miller, K. Stafford, J. Seymour, D. Light, W. Restetta, and D. Estelle. Thank you for the update. Thank you for the milestone note, courtesy and gratitude to them. Theoretically, prokaryotes that, before that, this publication was in Science 230, 144 to 149, year 1985. Right. Theoretically, prokaryotes that produce specific metabolites can be genetically manipulated in two different ways. First, their activity or amount of one or more of the enzymes and a pathway encoding the synthesis of a particular metabolite can be modified so that the amount of metabolite that the bacterium produces is increased. Second, foreign genes that produce enzymes that can use an endogenous metabolite as a substrate for the production of another metabolite not normally produced by the host bacterium can be introduced. While these sorts of manipulations are easy in theory, it is not necessarily easy to isolate and manipulate the required genes or to establish the appropriate conditions that enable complex biosynthetic pathways to function properly. To create a bacterium that synthesizes 2-keto-L-gulonic acid, which is the immediate precursor of commercially synthesized vitamin C, Anderson et al. Isolated the Corynebacterium gene encoding the enzyme that converts 2,5-diketo-D-gluconic acid to 2-keto-L-gulonic acid and transferred it to an urban sp bacterium that synthesizes 2.25-diketo-D-gluconic acid from D-glucose. The isolation of this gene was difficult because the enzyme had not been previously studied to any great extent. Therefore, before the gene could be isolated, the protein had to be purified and partly sequenced so that DNA hybridization probes based on the amino acid sequence of the protein could be designed. This work is an early example of what some workers have come to call metabolic engineering, which entails taking the genetic information for part of a metabolic pathway from one organism and transferring it into another organism to create a novel metabolic pathway. Now that can be analyzed biochemically, it was found that 130 kilodalton precursor protein is rich in serine, threonine, lysine, proline, and tyrosine. 60 to 70% of the amino acid contains a hydroxyl group. Most of the proline residues are hydroxylated to either 3 or 4-hydroxyproline hype, and the majority of the tyrosine are hydroxylated to 3,4-dihydroxyphenylalaniline, DOPA. Amino acid sequence analysis of the precursor protein further revealed that it is composed largely of repeating units. It consists of a decapeptide with a sequence as shown here. 7 of these 10 amino acids are hydroxylated. The cDNA for the 130 kilodalton precursor decyl protein was isolated from a cDNA library that was constructed with messenger RNA, mRNA isolated from the gland that actively secretes the bisyl adhesive. Both the adhesive protein and the cDNA have unusual features that might make cloning, expression, and production of a functional adhesive protein difficult in a heterologous host. First, the highly repetitive nature of the adhesive protein cDNA can make it unstable as a result of homologous recombination and subsequent loss of portions of the clone sequence. Second, proline, lysine, and tyrosine represent about 70% of the amino acids of the protein. Therefore, very high levels of synthesis may not be achievable because the corresponding intracellular amino acid transfer RNA tRNA pools might be limiting. When either complete or partial cDNAs for the adhesive protein were cloned onto yeast expression vectors and introduced into yeast cells, active novel forms of the adhesive protein ranging from 20 to 100 kilodalton were synthesized and represented a significant fraction, 2 to 5% of the total cell protein. Thus, there were no problems concerning either the stability of the cloned cDNA or the production of moderate amounts of the adhesive protein. Considerably high expression levels were attained. And when a chemically synthesized adhesive protein gene sequence was expressed in E. coli, in this case repeating gene units that encoded the consensus decapeptide repeat of the adhesive protein were used to construct a 600 base pair BP synthetic gene that encoded protein with a molecular mass of approximately 25 kilodalton. The 30 base pair repeat, the fundamental building block of the synthetic gene, consisted of codons optimized for E. coli expression. The synthetic gene was expressed at very high levels by using the T7 promoter. Notwithstanding the level of expression of the adhesive protein, most microorganisms are limited to the abilities to hydroxylate amino acids post-translationally, so the final protein might be under-hydroxylated. In fact, a number of tyrosine residues of the protein were not converted to DOPA, an outcome that limits the ability of the protein to form crosslinks. This deficiency was overcome by creating an in vitro hydroxylation system that used a bacterial tyrosinase in the presence of ascorbic acid to hydroxylate the tyrosine residues to DOPA. Ascorbic acid was included in the reaction mixture to prevent the premature oxidation of the DOPA residues to O-quinone. Oxidation must be controlled because it leads to cross-linking of the adhesive protein subunits. Like many other adhesives or glues, the protein adhesive must not be activated, cross-linked before its actual use. Antibiotics. Since the discovery of penicillin in the late 1920s, more than 12,000 antibiotics with different specificities and a variety of modes of action have been isolated from various microorganisms. The universal use of antibiotics to treat bacterial disease has resulted in an enormous improvement in human health and has undoubtedly saved millions. And the majority of most important antibiotics has been isolated from the gram-positive soil of bacterium. Streptomyces, all the fungi and other positive and gram-negative bacteria are also sources. The antibiotic market is driven by sales of 4 leading drug classes: the cephalosporins 27%, macrolides 20%, quinolones 17%, and penicillin 17%. Together, these 4 drug classes account for more than 80% of global antibacterial sales. So, an estimated 200 to 300 new antibiotics are discovered each year, primarily through labor-intensive research programs in which many thousands of different microorganisms are screened to find those that produce unique antibiotics. However, with the high cost of development, clinical testing, only the compounds that show significant therapeutic and economic promise are marketed. Therefore, only about 1 to 2% of newly discovered antibiotics are added annually to the disease-fighting arsenal. In fact, the pharmaceutical industry has been reluctant to invest in research and development in this area, and many companies have either abandoned or scaled down their efforts since 1999. In addition, to date, nearly all of the genetic improvements to industrially important antibiotic-producing strains have been achieved by the use of classic mutagenesis and selection, while the yields of antibiotics from many strains have been significantly improved. The original penicillin-producing fungus, isolated by Alexander Fleming, yielded 2 units per milliliter of culture, while strains used today synthesize approximately 70,000 units per milliliter of culture. This yield improvement took many years and required the use of considerable manpower and financial resources. Recombinant DNA technology can have a positive impact on this endeavor in 2 ways. First, the technology can be used to develop new, structurally unique antibiotics with increased activities against selected targets and decreased side effects. Second, genetic manipulation can be used to relatively rapidly and inexpensively enhance yields and hence lower the cost of production of existing antibiotics. For the generative manipulation of streptomyces, it is essential that it can be transformed and that the transformed cells can be readily selected. However, unlike E. coli, streptomyces strains do not exist as individual cells but as extended aggregates called mycelial filaments. The cell wall must be removed to release individual cells, protoplasts, before DNA transformation. Without this step, it would not be possible to distinguish transformed from non-transformed cells. because visible colonies on a solid medium would each have started from a cell aggregate rather than from an individual cell. Thus, colonies that grew in the presence of a selective antibiotic would contain a mixture of transformed and non-transformed cells. However, as a consequence of protoblast formation prior to transformation, all colonies that grow in the presence of a selective antibiotic contain only transformed cells. The uptake of plasmid DNA into streptomyces protoplasts is enhanced by polyethylene glycol. Following transformation, the protoblasts are first plated onto a solid medium to enable the cell walls to regenerate and are then overlaid with a selective medium that often contains either neomycin or thiostreptone, both of which act as selection agents for transformed cells. Cloning antibiotic biosynthesis genes. The biosynthesis of an antibiotic may include 10 to 30 separate enzymes catalyzed steps, so cloning all the genes for the synthesis of a particular antibiotic is not an easy task. One strategy for isolating the complete set of antibiotic biosynthesis genes consists of transforming one or more mutant strains that are unable to synthesize the antibiotic with DNA from a clone bank, constructed from wild-type chromosomal DNA. Following the introduction of the clone bank DNA into mutant cells, transformants are screened for the ability to produce the antibiotic. Then the plasmid DNA from the clone that supplies a functional gene and a gene product that complements a mutant strain is used as a DNA hybridization probe, to screen another clone bank of a wild-type chromosomal DNA, that is one in which the average size fragment is around 10 kb, to isolate clones with regions that overlap the probe sequence. In this way, DNA segments that are adjacent to and usually bigger than the initial complementing DNA can be identified and cloned. A complete gene cluster can be reconstructed from a single site on the chromosomal from the overlapping clones. If the antibiotic system biosynthesis genes are clustered at a single site of the chromosomal DNA, the genes that are adjacent to the complementing gene are also likely to be involved in the biosynthesis of the target antibiotic. However, if the antibiotic biosynthesis genes are scattered in small clusters at different chromosomal locations, it is necessary to have at least one mutant per gene cluster to obtain a DNA clone that could be used to identify the rest of the genes in the cluster. The complementation approach has been used to isolate some of the genes for the biosynthesis of the antibiotic undecylprotegiocin from Streptomyces pseudocolor A3. In this case, the complementation assay is simple and entails scoring the color of the colonies. Colonies of wild-type organisms are red because of the presence of antibiotic, and mutant colonies are cream-colored. Complementation produces a red colony. In addition to cloning antibiotic systems, genes by antibiotic biosynthesis genes, by complementation, more direct strategies can be employed. One or more of the key enzymes as a biosynthetic pathway can be identified through either genetic or biochemical studies and then purified. The N-terminal amino acid sequence of the enzyme can then be determined, and with this information, oligodeoxyribonucleotide probes for the gene can be prepared. This approach has been used to isolate the gene for isopenicillin N synthetase from Penicillinum chrysogenum. This enzyme catalyzes the oxidative condensation of the compound delta L-alpha aminodibyl L-cystaneal D-valine to isopenicillin N, a key intermediate in the biosynthesis of penicillins, cephalosporins, and cephamycins. Despite the difficulty, there are a number of examples of the cloning and transfer of large fragments of DNA encoding antibiotic synthetic pathways. In these cases, it is usually necessary to use a vector that can accept and maintain pieces of DNA as large as 100 kb. For this purpose, researchers have employed bacterial artificial chromosomes that have been engineered replicate autonomously in E. coli when they introduce streptomyces to integrate into chromosomes. Modulating gene expression in streptomyces. Streptomyces. To improve the productivity of antibiotics produced by Streptomyces spp, it is desirable to utilize a regulatory expression system that can suppress the expression of the target genes until the culture reaches the high cell density. It would also be beneficial if the system could be induced simply and inexpensively, and if it could function in a range of different Streptomyces spp. One regulatable high expression Streptomyces system that was recently developed utilizes the nitrulase operon from the Actinomyces rhodochrus. Excuse me. Let me repeat that for you. Rhodococcus rhodochrus. In this bacterium, the expression of the enzyme nitrulase encoded by the NIT-A gene under the control of the NIT-A promoter is positively regulated by the protein NIT-R, encoded by NIT-R, which is present in the same operon as NIT-A. NIT-R forms a complex with the inducer epsilon caprolactam before the complex binds to the NIT-A promoter, activating the synthesis of both proteins NIT-A and NIT-R. However, to use the system as part of a high-expression vector, a transcription terminator was placed upstream of the NIT-A promoter to prevent transcriptional read-through from other genes. A multiple cloning site for inserting target DNA was placed downstream of the NIT-A promoter, downstream of the multiple cloning site and target gene. The synthetic operon ends with another transcription terminator, and finally the vector contains the NIT-R gene under the control of the NIT-A promoter in a separate operon. When this expression vector is used upon the addition of an inducer epsilon caprolactam, the inducer NIT-R protein complex activates the transcription of both the target genes and NIT-R. The increased level of NIT-R results in a very high level of production of the target proteins. The system is still at a relatively early stage of development. With some target operations, it has been possible to achieve expression levels as high as approximately 40% of all soluble proteins. Moreover, the system functioned well when it was introduced into Escherichia, Streptomyces avermitilis, and Streptomyces griseus, bacterial strains that have all previously been used for antibiotic production. However, it still remains to be demonstrated that this expression system can be used as an effective method of increasing the yield of a commercially important antibiotic that is produced in Streptomyces strain. Synthesis of novel antibiotics. New antibiotics with unique properties and specificities may be produced by genetic manipulation of the genes involved in the biosynthesis of existing antibiotics. One of the experiments in which a novel antibiotic was produced, researchers began by examining the consequences of placing two slightly different antibiotic production pathways into one organism. Excuse me. A Streptomyces plasmid PIJ2303 carrying a 32.5 kilobase pair fragment of S. Seeley color chromosomal DNA contains all of the genes encoding the enzymes responsible for the biosynthesis of the antibiotic actinorodeine, starting from acetate. This antibiotics is a member of the family of antibiotics called isochromanoquinons. Isochromanoquinons, the intact plasmid and various subclones carrying portions of the 32.5 kilobase pair DNA fragment, example PIJ2315, were introduced into either Streptomyces sp strain AIIMS&NEET, which produces the related antibiotic medicine, or Streptomyces valsicirubra, And now the intact plasmid and various subclones carrying portions of 32.5 kilo base pair DNA fragment, example AIIGA-20-15, were introduced into either streptomyces sp strain AM7161, which produces the relative antibiotic metamycin, or streptomyces virus ruba, or B1140 or Tu22, both of which produce the antibiotic granitacin and dihydrogranitacin. Each of the antibiotics, actinidine, metamycin, granitacin, and dihydrogranitacin, functioned as an acid-base indicator, conferring on a growing culture a characteristic color that depends on the pH of the medium. The pH and color, in turn, depends on compounds being synthesized, mutants of the S-codicolor or aceticolor parental strain that are unable to produce actinidine or colorless. The appearance of new colors, in some cases following the transformation of streptomyces sp strain AM7161 or S. violacea ruba or S. violacea ruba Tu22, with a plasmid carrying either all or some of the genes encoding the enzymes that synthesize actinidine, suggests that a novel antibiotic has been produced. Streptomyces sp strain AM7161 and S. violacea ruba B1140 transformants contain PIG2303, produce the antibiotics encoded by both the chromosomal and plasmid DNAs. However, when S. violacea ruba Tu22 is transformed with PIG2303, a new antibiotic, dihydrogranitidine, is synthesized along with actinidine. When streptomyces sp strain AM7161 is transformed with PIG2315, a second new antibiotic, metarodine A, is produced. The new antibiotics represent minor structural variants of the pre-existing antibiotic actinidine, metamycin, granitacin, and hydrogranitacin, and probably arise when an intermediate compound from one biosynthetic pathway acts as a substrate for an enzyme from another pathway. As the biochemistry of various antibiotics biosynthetic pathways has been better understood, it has become possible to design unique antibiotics by genetic manipulation of the genes encoding the relevant enzymes. Engineering polyketide antibiotics. The term polyketide defines a class of antibiotics that are synthesized through the successive enzymatic condensation of small carboxylic acids such as acetate, propionate, and butyrate. Polyketide drugs include the antibiotic erythromycin, the immunosuppressive drug FK506, and the cholesterol-lowering drug, lovastatin. What previous polyketides are produced by plants and fungi, the majority are produced by actinomycetes as secondary metabolites. To create new polyketide antibiotics, the functioning of the enzymes that synthesize these antibiotics must be understood before the genes encoding the enzymes can be manipulated. Polyketide antibiotics are synthesized by a complex enzymatic mechanism analogous to that used for synthesis of long-chain fatty acids. Each condensation cycle results in the formation on a growing carbon chain of a beta-keto group. Polyketide synthesis consists of a number of steps that are each repeated several times, including keto reduction, dehydration, and enol reduction of the beta group of the growing polyketide chain. There are two classes of polyketide synthesis that are responsible for the synthesis of polyketide antibiotics. The synthesis that catalyze the biosynthesis of aromatic polyketides make up one class, and consist of one polypeptide with an active site for each successive reaction. The second class includes synthesis that assemblies of several polypeptides that have separate and distinct active sites for every catalyzed step in polyketides biosynthesis. These enzymes have a number of different domains, regions A through E, and each domain has a separate enzymatic activity and active site catalyzing a particular step in the process. The complete synthesis of a polypeptide antibiotic generally requires the participation of several of these multifunctional enzymes. Together, they make up the subunits of the polypeptide synthase. If each of the enzyme activities that is catalyzed by a domain on a multifunctional polypeptide synthase subunit catalyzes only a single subchemical biochemical step in the pathway, the loss of any one activity should affect only a single step in the overall synthesis. Moreover, alteration of a catalytic domain, whose function has been established should allow researchers to make predictable changes to the structure of the synthesized antibiotic. For example, a detailed knowledge of the genetics and biochemistry of the components involved in the synthesis of the antibiotic erythromycin allowed researchers to alter the biosynthetic genes in a predetermined manner and to produce predictably alternative derivatives of erythromycin. Erythromycin is synthesized by Saccharopolyspora erythrea and the entire 56 kilobase pair DNA segment that contains the ERY gene cluster has been sequenced. The erythromycin polypeptide synthase was altered in two different ways. Either one, or DNA region that encoded beta-ketoreductase activity was deleted, or two, a DNA region encoding an oil reductase was mutated with the beta-ketoreductase deletion. The erythromycin intermediates accumulated that had a carbon moiety rather than a hydroxyl group at the C5 carbon of the ring. Similarly, with the enol reductase mutation, a carbon-carbon double bond was introduced at position C6 and C7 of the ring. These experiments indicate that once the cluster gene encoding the biosynthesis of a particular polypeptide antibiotic has been isolated and characterized, it is possible to, by altering specific DNA fragments, to modify an antibiotic biosynthesis and pathway and thereby to alter the structure of the antibiotic in a predictable manner. Moreover, it is possible to cut and splice DNA fragments and thereby shift polypeptide synthase domain around to create novel polypeptide antibiotics. The gene clusters for aromatic polypeptides all contain a set of 3 genes encoding a so-called minimal polypeptide synthase. Each minimal polypeptide synthase contains the activities for one cycle of polypeptide chain elongation. The minimal module has a ketosynthase with an actual acyl transferase domain, a chain length factor, and an acyl carrier protein. The minimal polypeptide synthase is responsible for the synthase of the aromatic polypeptide backbone. Modifications to the basic structure catalyzed by other enzymes acting in concert with the minimal polypeptide synthase. The order of the modules in a polypeptide synthase specifies the sequence of the distinct 2 carbon units, and the number of modules determines the size of the polypeptide chain. The genes encoding a complete set of these proteins are generally organized into a small cluster. Each minimal polypeptide synthase gene cluster encodes the synthesis of a particular antibiotic. By interchanging genes between clusters, new aromatic polypeptide antibiotics have been created. This experiment demonstrates the potential of using genetic manipulation to design and produce novel aromatic polypeptide antibiotics. This approach promises to dramatically accelerate the process of discovery of new antibiotics. Improving antibiotic production. In addition to being a means of developing new antibiotics, genetic engineering can be used to enhance the yields and rates of production of known antibiotics. The large-scale production of antibiotics by streptomyces spp is often limited by the amount of oxygen available to the cells. The low solubility of oxygen in aqueous media, combined with highly dense nature of filamentous streptomyces cultures, often results in an oxygen-depleted culture medium, condition that causes poor cell growth and reduced antibiotic yield. To overcome this problem, it may be possible to improve the design of the bioreactors that are used to grow antibiotic-producing streptomyces cultures and to develop by genetic manipulation streptomyces strains that are better able to utilize the available oxygen. These two approaches are not mutually exclusive. One strategy used by some aerobic microorganisms to cope with oxygen-poor environments is to synthesize a hemoglobin-like molecule that can sequester oxygen from the medium and then deliver it to the cells, for example, the aerobic bacterium Vitrocilla, producing a homodimeric heme protein that is functionally similar to eukaryotic hemoglobin. The gene for the Vitrocilla hemoglobin was isolated and subcloned onto a streptomyces plasmid vector. Following expression of the Vitrocilla hemoglobin gene, S. cala, the Vitrocilla hemoglobin represented approximately 0.1% of this total cellular protein, and even though the expression was controlled by the native Vitrocilla hemoglobin gene promoter rather than by streptomyces promoter when both transformed and non-transformed S. cultures were grown in the presence of a low level of dissolved oxygen, that is approximately 5% saturation. The transformed cells with a functional Vitrocilla hemoglobin produces 10 times more actinorotin per gram dry weight cells and had greater cell densities than did the non-transformed cells. The expression of the Vitrocilla hemoglobin gene in oxygen-starved microbial cells may provide them with a general mechanism for obtaining sufficient oxygen to allow proliferation under otherwise limiting conditions. The compound 7-amino acid aminocephalosporanic acid 7-ACA is synthesized chemically from the antibiotic cephalosporin C and is used as starting material for the chemical synthesis of a number of cepham-type antibiotic cephalosporins. Cephalosporins have a few toxic effects on humans and protect against many different bacteria. Unfortunately, there is no known organism that can synthesize 7-ACA. However, a novel 7-ACA biosynthetic pathway has been constructed in the fungus Acrimonium chrysogenum, which normally synthesizes only cephalosporin C. The genes involved in this novel engineered pathway consist of a cDNA that encodes D-amino acid oxidase, and that comes from the fungus Fusarium solani, and genomic DNA that encodes cephalosporin acylase and comes from the bacterium Pseudomonas diminuta. Both of these genes were subcloned separately onto an acrysogenum plasmid expression vector under the control of a chrysogenum promoter. The first step of this new pathway, cephalosporin C, is converted into the compound 7-beta-5-carboxy-oxopentanamidol cephalosporanic acids keto AD7-ACA by D-amino acid oxidase. Some of this product reacts with the hydrogen peroxide, that is a byproduct of the reaction, to form 7-beta-4-carboxybutanamidol cephalosporanic acid, GL7-ACA. Cephalosporin C keto AD7-ACA and GL7-ACA is hydrolyzed by cephalosporin acylase to form 7-ACA. However, in the absence of D-amino acid oxidase step, only 5% of the cephalosporin C is converted to 7-ACA. Therefore, both enzymes are essential for high yields of 7-ACA. Although the level of 7-ACA that could be produced using the system was not sufficient to make this work the basis of a commercially viable process, it nevertheless demonstrates the feasibility of producing 7-ACA biologically. Medically important cephalosporins may be synthesized from either 7-ACA or the related compounds amino D-acetoxy-cephalosporanic acid, 7-ADCA, and disrupting the functioning of the acrysogenum CEF-EF gene results in the accumulation of large amounts of penicillin N. Moreover, when this mutant strain is transformed with the CEF-E gene from Streptomyces clavoligerus, penicillin N is converted to D-acetoxy-cephalosporin C, DAOC, which can then be converted to 7-ADCA. At the stage of the development of this process, not all the penicillin N is converted to DAOC. However, it may be possible to further increase the expression of the CEF-E gene. Thus, it is responsible to expect transgenic acrysogenum can eventually be engineered to produce large amounts of 7-ADCA. Many antibiotic producing organisms are slow-growing, require special growth conditions, or yield only small numbers of cells. To overcome these problems, E. coli was engineered to produce polyketide antibiotics at rates that are potentially useful for drug production. To do this, 3 genes, each 10 to 12 kilobase pairs in length, encoding the components of the polyketide synthase from the bacterium S. erythroia were expressed in E. coli, then a synthase from the bacterium then a Bacillus subtilis gene that produces an enzyme that attaches the cofactor phosphopentathione to the polyketide synthase was cloned into the engineered E. coli. In addition to supplying the polyketide synthase, there are sufficient building blocks, propionyl CoA and methylmalonyl CoA for polyketide synthesis. The E. coli gene encoding an enzyme that breaks down propionyl CoA was inactivated, and an S-C-Lacala gene for propionyl CoA carboxylase was introduced. Given the relative ease with which E. coli can be genetically manipulated and then grown in large-scale culture, this work may be significantly made a significant breakthrough. This work may be a significant breakthrough for the development and production of new antibiotics. Okay, now we're on to design antibiotics. In recent years, there's been enormous proliferation in the prevalence of antibiotic-resistant bacterial infections. At present in the US, more deaths are attributable to infections with methicillin-resistant strains of Staphylococcus aureus than to human immunodeficiency virus, or AIDS. And now on to rubber, natural rubber. The rest of it has been explained already. Cis-1,4-polyisoprene is an extensively used biopolymer that is obtained from a large number of different plants. The biosynthesis of rubber starts from simple sugars and requires approximately 17 enzymes, catalyzed steps, with the final step being the polymerization of isopentenyl pyrophosphate onto an allylic pyrophosphate. The last step is catalyzed by the enzyme rubber polymerase. Studies have been undertaken to determine whether rubber can be synthesized by genetically engineered microorganisms. As an initial step in this direction, a cDNA library was constructed by using mRNA from the rubber-producing plant Hevea brasiliensis. This library was then screened with a short synthetic DNA hybridization probe whose sequence was based on the amino acid sequence of a portion of the rubber polymerase enzyme. Antibodies directed against the purified enzyme were used to prove unequivocally that the cloned cDNA expressed rubber polymerase. This cDNA clone can now be used possibly in concert with other genes in the rubber synthesis pathway in an attempt to produce natural rubber in a microbial system. Alternatively, it can be used as a source of rubber polymerase to develop an in vitro catalytic system. In either case, research that might lead to a new synthetic route for the production of rubber is underway. Polyhydroxyalkanoids. This lecture will be over in a minute from now, and we will be continuing in the next lecture. Right? And polyhydroxyalkanoids are a class of biodegradable polymers that are produced by a number of different microorganisms, most notably alkaligens, eutrophs, and used as an intracellular carbon energy storage material. These compounds have thermoplastic, elastic properties depending on the polymer composition and are being considered for use in the synthesis of a range of biodegradable plastics. It's been estimated that by 2012, the US market for biodegradable plastics will be around $1 billion per year. Poly-3-hydroxybutyric acid is the most thoroughly studied and characterized polyhydroxyalkanone. Both polymers and the eutrophos genes encode its synthesis have been characterized. Poly-3-hydroxyvalerate, hydroxybutyric acid, copolymer poly-3-hydroxybutyrate 3-co-3-hydroxyvalerate, and another polyhydroxyalkanoid are produced commercially in the UK by fermentation of a eutrophos. That ends the lecture here. Thank you.